Welcome to the 7th class of this protein engineering course based on artificial intelligence, and this time we're going to review the topic of protein evolution. Remember that this is only the 7th class of a total of 10 that make up this course, which is completely free and available on this YouTube channel, so you can delve deeper into each of the specific topics. And before starting the class, the announcement is that the images, the quotes, as well as the recommended literature for each specific topic being addressed, will be found in the notes section of each of the slides. So for more information about the course, please visit its official website. Likewise, check the associated repository where you will be able to find tools, databases, as well as other courses on computing, artificial intelligence, among other topics, to be able to download the slides or implement them for a course you are working on. You can do it from the Google Drive or Zenodo platforms. To ask a question, you can leave it here in the comments section of the video on YouTube, or you can send it via any of my social networks, where you will find me as the user at Myinguar. And finally, if you found this course productive for your professional development and find it relevant, you can make a financial donation via PayPal in the following link. So let's review what topics we're going to cover in this session. There are 4 big themes, and the first is related to trying to become familiar with the multiple levels of biological organization, as well as the flow of information that occurs at each of them. The second topic will be related to biological evolution, to understand what is known as the RNA world hypothesis and its subsequent development to what would be the first proteins on that primitive Earth and how they diversified. Third topic, it will be a key concept that helps us better understand how proteins evolved, which is called sequence space, which practically consists of understanding the effect of mutations on this type of biomolecules. And as a fourth point, we will review how the interactions between proteins, as well as with their environment, model what this evolutionary landscape of these molecular entities is. So let's first review what the multiple levels of biological organization are. And before we can address all of these topics in detail, and also considering previously reviewed topics such as protein structure and function, 2 big disclaimers need to be made. The first is that evolution really lacks full agency. And the second is that when we usually explain some evolutionary phenomenon, we usually refer to a specific object, when in reality, what is evolving is the entire system, not just that part, although we obviously have to reduce it to be able to explain it in a more understandable way. But what evolves is the entire system. And here the idea is that usually when we tend to explain things, we use teleological terms, which is this idea that, well, things or events have some purpose to really explain their existence or cause. When in reality, perhaps in evolutionary terms, the most sensible explanation for a large majority of phenomena is really like this meme that perhaps you will already locate if you are from Mexico. But it simply happened, the right circumstances were given so that everything could be assembled together with the environment, and it just happened. That would be one of the most accurate explanations that at least I have found throughout my professional career. And this is not only something that I think, but they are also opinions shared by multiple colleagues around the world. For example, here we could find John Jumper's opinion regarding the evolution of proteins. John Jumper is in fact one of the authors of Alpha Food, which tells us that proteins would actually function as a kind of programmer that doesn't really know what it is doing. They have no purpose as the designed world of machines by us actually has. And despite all this, there is a great diversity of this type of biomolecules. As another opinion, we have that of a great evolutionary biologist called Antonio Lescano, whom I admire intellectually and who precisely suggests to us that life is the result of the evolution of the system as a whole. So when I tend to explain some concept here, as well as the explanations that I previously addressed, these 2 aspects must be considered, even though perhaps I also tend to be able to explain precisely this type of ideas, but in a teleological way. Another central idea that must be considered is how fragmented the biological record is, and from which we precisely construct our inferences in order to be able to do so. Create hypotheses about how evolution would have occurred, and then infer multiple other data. And this is very well reflected in what would be one of Charles Darwin's chapters on the origin of species, which precisely tells us that he really sees the geological record as a history of an imperfectly preserved world written in multiple, ever-changing dialects. We only have a single volume of that history, which only talks about 2 countries or 3 countries at most. And after this volume, we only have 1 chapter that has been preserved, which is quite short compared to the other chapters. And then within that chapter, each page only has a few lines there that have been preserved, and with which we can build the story. So when we think about this in biological terms, it could be reflected in terms of this tree made of proteins, where what we are seeing in terms of current biodiversity would only be the outermost branches of what this tree would be, and from them we try to infer what the entire evolution procedure of this type of biomolecules would have been like based on this idea of the primitive Earth. And well, that is the information we have, and it is the best we can do to explain this entire evolutionary phenomenon. And now we proceed to explain what the world of proteins is, something that is truly amazing in terms of understanding how these biomolecules are capable of forging our reality. They practically allow us to receive a multitude of stimuli that represent what we understand as reality, and that obviously also applies to other living systems, such as here the proteome of one of the bacteria, which would be Mycoplasma gentium, which is made up of only approximately 470 proteins. It is interesting that there will be some proteins that are located precisely in the cytoplasm, others that will be extracellular, or that will be embedded in the membranes. Its functional diversity is very considerable, and also its structural diversity. And it is interesting to observe precisely what the proteome of other types of species is like. What conditions made? In some cases there were 400 proteins, and in another quite interesting case, such as that of this archaea, which is in fact a symbiont, called Sachinarchaean mirabile, it turns out that its biology is composed of approximately 189 proteins, directly encoded by its genome. Obviously, making a symbiont depends on every other biological system independent of it, rather dependent on it, which in this case configures its reality in these terms of proteins. And when we look at how many proteins make up the human genome, the answer is a who knows, because there are multiple protoforms, which is really an enormous diversity of proteins. But when we look at it in terms of the genes that code for proteins, we could have an estimate that the human genome covers about 20,000 proteins. And here in this image, we are simply seeing around 16,500, that is, almost like 99% of them. And thanks to them we can perceive reality, talk, share ideas, and even give courses on protein engineering. The idea is that, really being a course on this topic, we are going to focus on this step of what is the multiple levels of biological organization, but in reality evolution occurs in each of them. It occurs at the level of DNA, RNA, proteins, metabolism, circuits, organs, organisms, and entire populations. And in each of these levels, evolution occurs precisely. And each of these levels, in turn, has certain limitations where precisely these limitations are practically forged by what would be its historical context in this sense. Phrase that must be considered is that precisely because we take in a historical record of everything that happened over time to arrive precisely at our existence, either as people or perhaps as an evolutionary lineage, right? For example, here we can review what would be considered major evolutionary transitions throughout the history of life. And what is relevant is to consider that each of these great transitions really involved an act of innovation in biological terms that starts from the reconfiguration of the pre-existing elements that were available and evidently new. Channels of genetic information are opened in this sense, where natural selection can precisely operate. Here on the left you see several of these evolutionary transitions, such as the formation of the first protocells, later the establishment of a standardized genetic code, then the emergence of the cells themselves, such as the eukaryotic cell, even the eukaryotic, and later the acts of symbiosis that would be like this integration of mitochondria, plastids, among other aspects, until moving on to what would be multicellularity. And quite interestingly, the establishment of what these animal societies would be, since they are actually constituted as societies, and that even we as societies, as evolutionary lineages, also have a mechanism that is quite characteristic, although perhaps not unique, which would be the language through which we can precisely share information. And that even now this new channel of information is also subject to its certain type of selection, such as cultural selection. The idea is that precisely in each of these evolutionary transitions, we really find multiple phases that allow their maintenance, as for example would be the idea of protocells, since now there are membranous compartments that allow the diversification of this type of protocells until what would be precisely the establishment of the cell itself. Later, for example, we can find other examples that when we reach a level of multicellularity, it is now also necessary to have a slightly more sophisticated regulation, to have communication between the multiple components that now constitute, for example, an organ or a multicellular system. And then in each of them precisely, there is also, well, a new type of information in terms of how it can be used, transmitted, and stored in this sense. For example, when we look at cells, it is evident that genetic information exists that can even be diversified through mechanisms such as sex. But when we look at what this evolutionary transition represented by natural language would be, we find that we now have a symbolic communication where we can share ideas beyond what genetic information would be. That they are even transmitted generation to generation, and that now these ideas, being symbolic, can be shared either through emojis, through writing, through music, even through the gesticulation of our face. We can share these types of ideas. The relevant thing about all this here is to understand that we as organisms are really historical structures that really reflect everything that happened before us in order to reach this point in our lives, in such a way that even natural selection is really a reflection of this entire historical process, and that when we try to explain any of these elements, we have to turn to analyze it retrospectively, because in order to give a certain explanation of how the entire process could have occurred to reach this specific point, here what we can observe are different evolutionary mechanisms. Natural selection is done, which is based on all of them, but that here we see that we can organize them into three large levels, each one being broader in terms of the spatial and temporal scales in which it would be operating. What we observe is that we can classify the first level as a level where variability is key. The second is a level where now this variability, in addition to being key, is joint with what would be the environmental conditions. And then this environment, evidently over many billions of years, now proceeds to a phenomenon where this part of biology is already beginning to make history. Because then now we are talking about contingent phenomena that could explain all the mechanisms that serve to diversify life forms. So for example, we start from what would be natural selection, and we can find small populations that will have intraspecific variations in this sense. And then thanks to the environmental conditions, we can begin to see precisely how this intraspecific variation is already beginning to establish itself as even regional variations, specifically associated with that specific type of environment. And over time, this is now spoken of in terms of macroevolution, where, due to this genetic diversification, we even speak that there are now phylogenetic limitations. That perhaps we will be able to see them reflected in the form of perhaps then a species of a certain lineage, since now it is no longer capable of reproducing with another species of a completely different lineage. The idea is that there are multiple evolutionary mechanisms beyond natural selection, and that they really operate at different time scales, although it is evident that natural selection is really the basis for all of them. And the idea is that precisely thanks to this type of information and reconfiguration, recombination, among many other things, all this information can be reflected within what would be the proteins, in such a way that in principle, sequence should reflect what the information of each of these levels would be. For example, when we inspect what the atomic information of proteins would be, we find that there is a specific arrangement regarding the isomers that make them up, right? The type of bonds or even the type of atoms that were in great abundance on the primitive Earth. But it turns out that only some amino acids were selected by some specific circumstances to be included in those that are proteins. So now we move on to this second level where we find amino acids that have a more polar, non-polar behavior, with a positive, negative, or aromatic charge, and that met some condition to be present in proteins. We find that it is thanks to these proteins and their reconfiguration, their interactions, that allow us to form these interaction networks that we now see reflected as metabolism. And now in turn, the information that we can find about proteins related to a specific type of metabolism, we can also relate to their cellular context, where we can now think about the microenvironments where these metabolic networks are found, as well as the cell types where we find ourselves, given that, for example, when we look at the type of cells that constitute us, we will find that we have cells that are neuronal, cells that are epithelial, cells that are from the stomach, among multiple other cell types that we have, which in turn now make up tissues, where now we can find what would be isoforms, which are expressed in a quite specific way for this type of tissue, which, for example, could be a genetic expression, particular of a protein in the liver and another, for example, in the heart. Later we now move on to the idea that this is really encompassed by what would be the idea of an organism, where we now have to analyze the organism in terms of its interactions with other species, as well as its environment in ecological terms, and that we can even also approach it from an evolutionary sense. And so then, we see all this protein information reflected now in spatiotemporal terms, whether on a scale of nanometers to kilometers, or even in terms of seconds to billions of years, to be able to understand the evolutionary phenomena related to this type of species. For example, when we look back at what atomic information would be, we can think about this key concept that is chirality, which is how atomic structures are arranged in this three-dimensional space. And very curiously, it turns out that most of the proteins that we find in nature, they turn out to be a type of isomer that is known as a rotary wolf, which is actually composed of a type of amino acids, an isomer, that has a configuration that is known as L-amino acids. Here what we see is precisely these two isomers, and we see that in this case the L-amino acid, when placed in front of a mirror, produces a mirror image that in this case is known as D-amino acids. And well, curiously, it turns out that throughout evolution, due to the conditions of the early Earth, we really do not know what circumstances happened. Perhaps what would be the L-amino acids were selected in favor to really constitute all the biodiversity of proteins that we currently observe. Yes, there are amino acids scattered around. They are even used by living systems. However, the vast majority is made up of what are L proteins. And so it is curious to look at this and think if throughout evolution, really all these types of interactions and mechanisms that are supported by what would be natural selection led to the development of this type of molecules. It would be possible that two entities, say in this case a D protein and an L protein, they could interact and establish new information channels, as previously occurred with the entire lineage of L proteins. That's something we don't really know, and it's obviously the task of evolutionary biologists to investigate whether, for example, this is possible. Although, well, that's actually how it is. We can also see that what stereochemistry is really matters, and matters a lot. And these are examples that are usually brought to the table to try to share the idea of its importance. Here what we find is a compound called limonene, which also has its respective isomers, which in this case we classify them based on how they divert the polarized ones. So we have one that is more limonene, and one that is called less limonene. And again, this goes in the direction of polarization. In what direction does it direct the polarized light in such a way that the positive limonene is called dextrorotatory, because this polarized light deflects it in a clockwise direction and vice versa, which would be the less limonene that is levorotatory, and then it would go counterclockwise. And it turns out that the behavior of these two molecules with respect to our senses, it is completely different, because what would be the most limonene we perceive as a smell associated with oranges, while the least limonene we associate precisely with a lemon smell. And it is quite interesting to consider how this effect is completely different, despite the fact that they are composed of the same atoms, but thanks to their three-dimensional projection in space, they have a completely different effect. Another type of isomers that is quite relevant is, for example, those associated with this compound called thalidomide, a medicine developed approximately between the 50s and 60s, which was intended to be applied to what would be neonates in order to help them with multiple health issues. But it turned out that when the drug was generated, the importance of stereochemistry was not really considered, because they were not topics that were really known at that time. So what was administered to the patients in the drug was its version of thalidomide. And it turns out that thalidomide results in at best a deficiency in the development of the limbs of individuals. It can even kill them in this sense, and there was a large, quite severe problem where approximately 10,000 children were affected with severe deformities in their extremities, which we already see now in this photograph where they are now. Adults and evidently cannot lead a common life like the rest of the people who had normal development, due to this poor execution of a clinical treatment. So the idea of this is that stereochemistry really matters, and it matters a lot. Well, their effects can be quite different, even though they are composed of the same type of atoms. But its three-dimensional configuration in space is quite different from its effects on us. Another aspect that we could consider in evolutionary terms is this idea of how a protein can fold into a three-dimensional folding pattern, and obviously then adopt its native form and execute its associated functions, whether structural, enzymatic, among many others. And then here exists precisely what is known as Levittel's paradox, where we can really think that this exploration of the folding pattern is not as random as we would think, since perhaps we could think about it now in terms of the hydrophobicity of this type of residue. For example, there are some residues that we already saw that are more hydrophobic, others that are more hydrophilic, and that precisely the idea that they remain buried because they are dissolved in this aqueous solution could promote the idea that we would have an alphabet of two large types of waste, right? And given this context, perhaps now we can diversify to what would be the 20 types of proteinogenic amino acids, and subsequently how their side chains are configured to give way to their biological activity. Another interesting aspect of proteins is the immense combinatorics that can exist based on the number of residues they contain. For example, we can think about how many numbers of possible sequences we can form from a total of 100 residues, which we are seeing here on the x-axis and the y-axis. And it turns out that when we talk about proteins with approximately 100 residues, we will find that the atoms that make them up would actually practically exceed the number of atoms present in the universe. And even when we think about proteins that have just 50 residues, we would be talking about them having more atoms than grains of sand on Earth. So how did evolution explore all these possible combinations? In order to find folding patterns that were useful in terms of biology and the context in which they were embedded. That is something that we can think and reflect on, since evolution really is a magnificent machinery to be able to find these types of solutions in biological terms. We can also reflect on how it is possible that it has been detected that there are more than 500 types of amino acids, and yet only 20 of them are the ones that we usually associate with precisely the formation of proteins. Why did that happen? There were certainly some conditions that favored precisely these amino acids to be selected. The idea is then that really in these conditions of a primitive Earth, there really was a bioavailability of this type of ingredients so that they could constitute the proteins that we now see reflected today. For example, in this primitive land, perhaps a central idea was that amino acids with hydrophobic behavior were the first to be integrated into proteins. Perhaps there was also at that time a kind of competition between the diversity of amino acids that exists with others that are considered non-canonical, since they are not integrated into this alphabet of 20 proteinogenic amino acids. And later, those that were considered the first amino acids were found with a little greater diversification. For example, now adding hydroxyl groups or with acidic behavior in what would be their side chains, thus constituting the first forms of proteins, which did not have large fractions of secondary structures formed. They were relatively disorganized at the time. Or we thought they were disorganized at the time. And later other types of amino acids began to appear. For example, those that already have positive or negative charges, that were aromatic, etc. Until we reach the versions of proteins that we can currently see, which already have a high content of secondary structures and quite sophisticated arrangements in terms of their symmetry or their quaternary structures. And here is precisely what we see, how these types of alphabets were created and how they expanded. It is thought that what was this first amino acid alphabet was mainly made up of these hydrophobic amino acids such as alanine, glycine, valine, which were actually part of the first proteins. Later it was extended to an alphabet of 9 residues, where what we were able to see is that precisely now there are these hydroxyl groups, which would provide the system with a certain reactivity due to the negative charge precisely of the hydroxide group. Subsequently, this alphabet was now expanded to one of 16 residues where we already found what would be these charged amino acids such as arginine, lysine, even cysteine. And finally, we arrived at this alphabet of 20 amino acids where we even already have precisely the presence of these aromatic rings, and which would also be contributing to what would be the folding of proteins. And this is precisely one of the hypotheses we have regarding why only 20 residues were selected to be present in the proteins. And it turns out that perhaps these 20 amino acids were selected due to their foldability. That is, their ability to fold, which would have been precisely a selection factor, a selection pressure that would explain why this is yes and the rest of more than 500 amino acids are not. And this is something that is partially reflected in what this experiment would be, which consists of generating 1,000 sequences of 25 residues that were assembled at random in this sense. So we have as ingredients different types of libraries, where different types of amino acids were used. And in each of them, obviously, we are now going to evaluate the degree of disorder. And the degree of order of its secondary structures is made of alpha helices or beta folds. So when we have a library of residues that were prebiotics, it turns out that these randomly formed proteins have a large, completely disordered content, and the fraction of secondary structures is relatively poor in this sense. Contrary to what would be the secondary structure fraction, when we created proteins at random, using the 20 proteinogenic amino acids, but removing what would be the cysteine residues. A practice that is usually relatively common when we want to make random proteins, because cysteines, as they tend to establish these disulfide bridges, they can cause aggregates that would be quite annoying in experimental terms, since they would not necessarily be stabilizing a native conformation, but would even be the formation of aggregates in this sense. And it is in fact this particular library that is now considered to have a higher structural content in terms of its secondary structures, when compared to other variants derived from this library composed of the prebiotic amino acids. For example, we can build a library based on these prebiotic amino acids, but now we replace the branched amino acids with their unbranched equivalents. And precisely by removing this branching property, we see that the content of secondary structures is decreased and the degree of disorder is increased even more. We can build another variant of this prebiotic library where we now add an amino acid, arginine. And remember that arginine It is a relatively modern amino acid. Let's call it that, because it used cationic behavior, and it does improve what would be a little, very little, which would be the structural content of its secondary structures, and the degree of disorder really seems relatively the same as the prebiotic library. We can even consider an amino acid that at that time was a prebiotic. It was, let's say, a neighbor. Ah no, sorry, I was wrong. A variant of the prebiotic library where precisely a prebiotic amino acid is added that also had a cationic behavior similar to arginine. And what we observe is that this condition occurs again, where there is no good formation of secondary structures, and the degree of disorder is promoted. And even if we add a residue with aromatic behavior such as tyrosine, well, what would be the degree of order is improved relatively a little, but not as much as if we considered all of the 20 proteinogenic amino acids, except for the behavior of cysteine. So the idea is that what branched amino acids are really what they allow is this hydrophobic packaging that perhaps could have been better adapted to be able to diversify in terms also of their cofactors that can bind, of the type of RNA that can exist, identify and also evidently recognize, join with it, as well as its capacity to be able to carry out enzymatic activities on this primitive Earth. Because even if we do not only consider precisely these unbranched amino acids, or those that have side chains that are relatively short and of a basic behavior that in principle were abundant on this primitive Earth, it turns out that we can find that the peptides formed from them are now oversolubilized, so that their packaging is actually quite bad. And this contrasts quite a bit with this idea previously raised, that precisely this alphabet of proteins we can see as two large types, the residues that are hydrophobic, and obviously understood this within a context such as the aqueous solution, where proteins are thought to have developed on the early Earth. The idea also here is that precisely throughout this primitive Earth, there was a change from chemical evolution to biological evolution through the interaction of this type of biopolymers. And a type of interaction widely suggested in the literature as a key factor in biological evolution was the interaction between proteins and what would be biological RNA, which even had space under precisely these conditions before the protein alphabet was properly established, as we already reviewed. Here the idea is that obviously we start from multiple biopolymers, multiple ingredients such as metals, cofactors, and obviously water molecules, and that little by little these peptides were formed or with their interaction with oligonucleotides, forming precisely these protein complexes with what would be the RNA, and eventually what would be these later amino acids already appeared where the interactions between this type of biomolecules were already established relatively well to form what would be the relatively modern proteins that we see today. And here the idea is that there is also a component that some consider relevant within this hypothesis of how they could have emerged that would be the conserved ones, which is the idea that thanks to the interactions established between what would be the RNA and the protein, the aggregation of this type of compounds is promoted in such a way that they now behave as different phases in aqueous systems. When seen with greater interaction between two types of biopolymers, there is a density that distinguishes them in the aqueous phases and in the biopolymer phase that begin to isolate them so that they could diversify their interactions in this sense. And obviously, there were also other mechanisms such as the interaction with minerals, with cofactors, in this case, for example, being able to establish an efficient catalysis, and not as basic as the catalysis capable of carrying out RNA, would be even the formation of these micelles, these first vesicles, these first membranous systems, such that there would now be a certain compartmentalization, until what would be precisely the emergence of this last common ancestor, now already composed almost precisely as a cell, as we usually think of it in this sense. The idea here is that we really start from multiple chemical precursors. There will be multiple paths where there can be precisely this chemical evolution, But in practically all of these cases, there will always be an interaction of RNA with protein. Here what we can see is precisely a diagram of this type of interactions between biopolymers, and that we can distinguish in three main pathways that are declared here in different colors: yellow, blue, or red. For example, this would be a path more focused on the development of RNA. This would be a more developed path to what would be protein synthesis, and this would be another that tries to cover both at the same time. So for example, we start from these canonical components, that is, the nucleotides, the nitrogenous bases, which eventually give way to the formation of RNA. And RNA, as it has catalytic capabilities, can carry out this somewhat elementary catalysis with certain elements available in the environment, in such a way that a pre-ribosome begins to be formed. Given that what is the ribosome, they can also adopt certain folding patterns, just as proteins do. And not so much like that, even what DNA is, which is thought to have been later than what RNA would be. And eventually, through the interaction of this peribosome with amino acids, we move on to what would be the formation of enzymes, even the establishment of the ribosome, and we arrive at a protometabolism, where now there is a great feedback between this type of molecular ingredients. And we now move on to see what this pathway would be, where it has greater emphasis on what would be peptide synthesis. Once again, we see the canonical ingredients, such as amino acids and nitrogenous bases. We move on to this idea of a pre-RNA that, with its interaction with the amino acids available in the environment, this is more of a hydrophobic nature. We move on again to the establishment of this interaction of RNAs with small peptides. And in turn now, we return to the idea of the pre-ribosome, the ribosome, diversification into enzymes, and again, we arrive at the idea of protometabolism. When we now consider the set of these two pathways, we can obviously speak equally again of amino acids and nitrogenous bases, each the same forming in this case increasingly complex peptides, now reaching an enzymatic capacity, which is also fed back by the idea of a peribosome, arriving again at the preparation of enzymes and the establishment of a pre-autometabolism in this sense. So it is quite interesting to think how it is that always in each case, this interaction between RNAs and peptides always emerges, which we think is one of the key hypotheses in what would be the development of life and the diversification of this type of biomolecules. Also here it is obvious to consider that this was not the only mechanism, but that there was even a coevolution that we also see reflected in what would be the establishment of this genetic code that we consider as a standard. For example, we also think that precisely this genetic code, due to the coevolutionary conditions in which it developed, let's say that it arose under selection pressure that minimized the deleterious effects of mutations, but also allowed protein diversification. In this case, we can see this effect of buffering mutations by finding how there are codons that are synonymous. We could see that there are multiple ways to be able, for example, to encode a tronine amino acid, which in this case we do have, to start from an adenine, then from a cytosine. But in this case, we have 4 possible combinations, so that they could encode what would be a tronine residue. However, the idea that the genetic code is standard, perhaps it can apply to a large majority of life forms, but in reality, we also find particularities that can be found regarding the genetic code that are used in certain cases. For example, we found that this genetic code standard, it is not so applicable when we look at other forms of life, and in particular, even what would be the mitochondria of other forms of life, such that then we have detected what would be up to at least 33 variants of the genetic code, where the codons that we see in this standard model do not necessarily correspond to the same type of amino acids. Here there was a reconfiguration, such that now they are codified. Perhaps now term codons for what would be a codon associated with serine among other aspects, and we find this type of variations in the genetic code, as you can see here in blue, what would be mitochondria or even plastics, which is interesting to observe when we think about the idea of symbiosis, since this encompasses the idea that there were 2 independent organisms that under the appropriate selection pressures, well, they were already integrated in such a harmonious way that now they can no longer live in each other. But it turns out that even at the level of the genetic code, we can see these differences that perhaps we do not see in other terms, such as metabolic integration or even the separation of what would be their cells. So now we are properly addressing what the evolution of proteins is seen in general terms in terms of biological evolution. Here this is the central idea of how proteins evolved, and we are going to cover it in a little more detail for each of the steps. The idea is precisely that there was this evolution of chemistry on a primitive Earth that establishes precisely this prebiotic chemistry. Later, these motifs appeared, these structural signatures that we already covered in the class of protein structures that turn out to be small fragments of secondary structures that are even present in folds that have nothing to do with it, that are completely different folds, and that curiously are also present in these superfolds, which we think could serve as nucleation centers during the folding process. So that these little structural motifs, these structural signatures, well, they were diversifying with respect to their dimensions, and obviously the mass, obviously gaining more and more ways to decorate them. Let's think about it like this. Arriving at the establishment of the first proteins or ancestral proteins, which we think had a very high degree of symmetry. As you can see here, we find homomultimers. In this case, they would be composed of 1, 2, 3, 4, 5 chains. And that perhaps these first proteins, being symmetrical, only had to practically duplicate themselves to establish slightly more sophisticated interactions, which eventually diversified or recombined to reach what would be the modern or relatively modern proteins that we observe, where there is already a recombination of secondary structure elements, forming proteins that are quite diverse in their shapes, as well as in the type of quaternary interactions that they can establish. And it is thanks to all this evolutionary understanding that we can apply protein design to look at the solutions that evolution found and try to develop them more specifically to a context that we are trying to design, how to perform a function or meet certain structural requirements to use as if it were a scaffolding. So now we move on to explore the idea precisely of these first peptides, or these primordial peptides. And to do this, we must understand precisely the contribution that the ribosome has in what would be the history of proteins. Because, as I mentioned, the ribosome is actually a ribosome, and when we cover it in classes, we really tend not to grasp this idea because the ribosome is usually presented in a schematic way with this type of diagrams that are easy to communicate the idea, but that perhaps do not represent the complexity that implies that the ribosome is a ribosome. What we see here is precisely the ribosome doing its translation procedure, and we see that the ribosome is composed of a larger subunit, which would be indicated with a 50S, and a smaller subunit, indicated with a 30S. Well, it turns out that the ribosome, if we take only in illustrative terms what would be the minor subunit, or the 30S, because really here it is much clearer how it is that the ribosome is a ribosome, where really the RNA that we can see here in blue, royal, and pink color, because it constitutes the majority of what makes up this molecular entity. Such that because the proteins that are associated with it tend to be in this sense a little more exposed on the surface, but it is thought that precisely what these protein structures do is contribute in structural terms to the proper folding of this ribosome, so that now it is this ribosome that is capable of precisely translating this sequence of mRNAs into polypeptide chains. So it is relatively consistent to think that when we make phylogenies of the tree of life, or phylogenies that aim to include multiple domains of life, such as the part of archaea, eukaryotes, or bacteria, since we can use this type of ribosomal proteins as one of the most conserved elements in terms of all forms of life, since obviously to exist it is necessary to have a ribosome in most circumstances, in most cases. And for example, we see that what would be this quite famous phylogeny of the year of life is precisely built with proteins associated with the ribosome that turn out to be conserved. In these three domains, there will be ribosomal proteins that are specific to each one, as you can see this set of proteins, that is characteristic for eukaryotes, this set of ribosomal proteins that are characteristic for bacteria, but here there is a common one of which we can find a certain evolutionary history shared between this type of elements, and this is then where we address the idea of the world of RNA, which suggests that this evolution of protein folding actually co-evolved with what was the development of the ribosome. And it is precisely what we are seeing in this diagram, where we find here an RNA molecule. Here we also have another protein molecule, and that throughout billions of years of evolution, since these two molecules were interacting, establishing increasingly sophisticated interaction, now duplicating in order to establish the structure or specific folding of this RNA, until now finding a macromolecular complex divided into two large subunits, such as the ribosome that we saw previously. And to be able to talk about the ribosome, a key idea to understand it is to think precisely of ribosomes as a kind of molecular onion, as proposed in this review, as is. We can understand the ribosome with the idea of layers in such a way that the proteins associated with what would be its center go to its most relevant part, which would be this peptidyl center, where the transfer is made to be able to generate precisely the proteins. There are proteins that are very well conserved, and that as we move away from this peptidyl transferase center, there are different types of proteins with different degrees of conservation. And that is why we can understand ribosomes as if they were onions, and these onions evidently seen from an evolutionary term of the type of proteins that were coupled to this type of macromolecular system. When we turn to see precisely what type of molecules can interact with what RNA is, we find that at the center of all possible interactions, we obviously find other RNA molecules. We find ribosomal proteins, and we also find inorganic cations, metals. And metals, as I mentioned, also have a great property, which is that in order to establish this type of interactions, they have to satisfy a fairly coordinated geometry, in such a way that precisely these tetrahedral, ultrahedral, anglisplanar formations are promoted, among other forms that really support in structural terms a greater diversification of interactions. As a second sphere of entities that we have that can interact with the ribosomes, again, with the RNA, we find, for example, what would be the transfer RNAs, which would now be the amino acids, or messenger mRNAs, among other aspects. And as a third phase of elements that can already interact with what is RNA, well now, we already find a great diversity of molecular entities, which for example could be processing tRNAs, chaperones, the proteasome, among many other aspects. The idea is then to understand that there really are many molecular entities that could interact with the ribosome in its early stages, and that thanks to these interactions precisely, there was a functional and structural diversification of the ribosome in such a way that we think of the ribosome in terms of its development over time, thanks to a model called the molecular accretion model, where precisely we see how the ribosome expands from small, independent ingredients to a fairly large and quite sophisticated macromolecular complex in terms of the structures and the functions that are performed. As a first point, we would find precisely that what are these first relatively simple foldings of RNA are formed, as well as the formation of helices similar to what would be the double helix of DNA, but in this case, it will be constituted only by RNA, since it is only a single-stranded helix. The second point of this accretion model is precisely the idea that thanks to these three-dimensional RNA patterns, it is now also possible to carry out the phenomenon of catalysis, and especially what is precisely this peptidyl transferase center found in the center of the ribosome. Finally, what would be? Well, not finally, in an intermediate way, would be precisely that now these subunits begin to gain mass. By diversifying, by joining precisely with other proteins that are also diversifying in turn, making this ribosome increasingly more complex to the point that it is now unthinkable that these two entities could not coexist precisely, establishing a coevolution between the ribosome, or rather the RNA, and the ribosomal proteins, and then other relatively similar steps where we find that the ribosome only expands in terms again of its dimensions of its mass, now incorporating what would then be protein domains with a greater fraction of secondary structures, until arriving precisely at the idea of a relatively modest ribosome like the ones we observe today, that are already composed of multiple peptide chains quite well structured, and that precisely establish the functionality of this entity to be able to generate proteins. Again, when we turn to look precisely at the degree of conservation of the ribosomal proteins in relation to each of these stages, we find that what would be this first layer of the molecular onion, this ribosome, it turns out that these ribosomal proteins are conserved in their sequence by around 90%, and that when we go to what they would be, the second layer, the third, the fourth, the fifth layer of this molecular onion, we found that there is even a quite considerable conservation of its sequences of around 60%, which is a reflection that since this is a fairly important activity in terms of proteins, due to multiple factors, it is selected in favor of being conserved throughout evolution, which is quite relevant given that when we talk about evolution, we are really talking about billions of years where multiple experiments could have occurred by evolution, and that perhaps many did not work, and that only now we find those that worked in these terms. And for example, here we can see precisely how it is that what would be this increase in the size of both the larger subunit and the smaller subunit occurred, in terms of what would be the comparison of evolutionary lineages that are most divergent from us. This does not imply that we are the most evolved forms. That would be a completely erroneous reading, but perhaps a more ad hoc reading would be precisely to know that we as an evolutionary lineage, since we have a time window that is much shorter than what it would be the emergence of evolutionary lineages such as the group of Epistasis, bilaterians, etc. So, when we turn to see what would be the increase in mass of what is the minor subunit of the ribosome, we find that these first forms associated with what would be archaea or bacteria have approximately 1,000 nucleotides in their RNAs. And it is even quite interesting how this subunit was not really gaining, well, a great diversification in terms of nucleotides, even despite coming to us as a more derived evolutionary lineage, right? But when we turn to look at what the largest subunit of the ribosome is, we find that, well, yes, archaea have, well, both archaea and bacteria have ribosomes of approximately 3,000 nucleotides, and that later when they meet, several appearances of these evolutionary lineages throughout the Epistasis, the bilaterians, the sarcopterygians, the now establishment of amniotes, even of what we would already be as mammals, we are precisely gaining nucleotides in these RNAs to the point of reaching our ribosomes that are approximately 5,000 nucleotides long. We do not have the largest ribosome detected. However, this idea helps us understand how the ribosome diversified in structural terms and also in functional terms of each of its elements. And so now here the idea is to consider what they might have looked like, what the first proteins looked like on this primitive Earth. Reflected here with this small illustration of precisely one protein on this completely chaotic Earth, with many circumstances that could be quite dangerous for a typical common cell, and yet the conditions were met for us to be here. So we return to the idea precisely of these structural signatures or these motifs which we think were key. During these first steps of the ancestral proteins, it turns out that when we analyze all the proteins as a whole, we can precisely find several of these motifs, several of these structural motifs or structural fragments that are present in all of them, which we think could represent an idea of remnants of this idea of the world of RNA that has a fairly strong interaction, as we already saw with what would be proteins, and that precisely these small peptides could have promoted the idea of, well, the folding of proteins given that they divide as nucleation centers. So here we see a vocabulary composed of 40 of these small structural motifs, that each of them is also characterized by the type of functions it can perform. For example, here these colored in yellow turn out to have considerable abilities to bind to both RNA and DNA molecules. And for example, this type of structural reasons, we could find them mostly present in what would be ribosomal proteins, given that precisely what is thought is that this type of proteins served as a kind of scaffolding, so that the ribosomes of the ribosome precisely adopted their three-dimensional structure in order to carry out catalysis. There will be some other structural motifs, which precisely have a greater tendency to associate with what would precisely be metal ions. And as we already covered, these metal ions also provide a certain specific geometry to bind, promoting a certain structuring of these molecular entities. And so we find different structural motifs, which, even though we are talking about completely different foldings, it turns out that when analyzed in structural terms they are shared, which suggests that perhaps when we approach protein evolution in terms of folds, perhaps it is worth exploring protein evolution now in terms of these structural fragments. And now we move on to review precisely how this type of fragments, represented here by this specific example, can be found in multiple folds or in multiple superfamilies. For example, this is one of the motifs called the elixipirin elix motif, which turns out to actually be part of multiple folds and multiple superfamilies. For example, here we can observe several superfamilies and how this motif is always found in these families, but in each of them there are evidently a greater number of alpha helices, completely different arrangements, now including folded betas. But when we take this specific motif, we find some conservation in terms of the residues that can configure it, as you can see here in this alignment, composed of several sequences related to multiple superfamilies, and it turns out that we always find patterns like this leucine, then this glycine, valine, again another glycine, etc., which really is a reflection that during these early Earth conditions there was a limited number of solutions that was adopted to precisely diversify on it, and that results now, as we can see precisely, as a remnant of these first primordial proteins, now present in multiple superfamilies here, is also the idea of understanding how this phenomenon of convergent evolution occurred in such a way that this could be a reflection of the number of limited solutions that were available at that time, be able to constitute what these catalytic arrangements are, and precisely carry out catalysis, or also promote the formation of certain three-dimensional structures. For example, during these first steps of life, it is thought that interactions with phospholigands were quite vital, given that precisely these types of phosphorus-based compounds are highly energetic, as we can see reflected in what is this almost universal currency for all forms of life, which is ATP or AT. And when we think about how many proteins are capable of carrying out this type of function or binding with phospholigands, we find that at least 275 times throughout evolution proteins capable of establishing interactions with phospholigands appeared independently. Something we see in this graph, which on the x-axis would be a representation of the number of families contained in each of these folds. And on the y-axis is how many of these families, that is, what fraction of these superfamilies are able to bind phospholigands. All of them bind to phospholigands, but evidently some have larger members, and these members also do so in greater quantities. And it turns out that when we turn to look at precisely those that have a high content of families, and that also that high content of families can mostly bind to phospholigands, it turns out that we found some fold that are considered ancestral. When we do this type of retrospective analysis, we find that these ancestral folds, such as the hubdomen, the flavodoxin fold, the Timborough fold, the Pillups, or the Rossman folds, all of them are quite skilled at being able to precisely bind to phospholigands, which, well, it is quite interesting when we look at their structures again, given that they are always composed precisely of a beta fold, then a turn region, and then an alpha helix, where precisely these phospholigands tend to join precisely in what is the commissure of the beginning of this alpha helix, which precisely also reflects a limited possible number of solutions that could be covered during this first period of protein diversification. So here precisely what they suggest to us is that perhaps considering the evolution of proteins in terms of domain could be a very large or thick representation, and that perhaps it would be worth turning to look at the proteins when we analyze their evolutionary history in terms of the fragments they present, given that it is quite curious how the ability to bind to phospholigands independently evolved 275 times, which is, as I mentioned, a very relevant energetic way in terms of life. We can also see evolutionary convergence reflected in what would be the architectures of the active sites, given that precisely there is also a specific three-dimensional configuration that has to be satisfied in order to carry out catalysis, as with ideas of precisely being a that has a tendency to donate or accept electrons, and effectively be able to proceed with what would be the formation or destruction of covalent bonds in this sense. And here we can approach this example of what convergence would be at the active site level using the example of carbonic anhydrases, which, as you can see here, there are several types of carbonic anhydrases. Here in this case only 3 are shown in this graph. They see that in some cases there is a great predominance of alpha structures, such as in this case a magenta one, another that is mainly made up of secondary structures of the folded beta type, or that is a combination of both alphas and betas, but it turns out that the active sites, when we align them, all of them have a quite similar mechanism with stidine residues and also coordinated precisely by a zinc ion. Something that is quite equally relevant to consider is that there was a limited set of solutions to be able to carry out precisely the catalysis of this type of enzymes. This is not the only example of convergence at the active site level. For example, we can now look at the serine proteases. Again, we see examples of how their foldings are completely different. But when we align their active site, we again find that it is precisely this same type of residue that is found in them to be able to carry out catalysis. So what this convergence of the active site really suggests to us is that there was a limited number of configurations capable of carrying out this biochemical reaction, and that precisely because this number of solutions was reduced, since it was a greater tendency to converge precisely to a single solution. And while it is relevant to consider the effect of these convergence phenomena on what protein biodiversity is, it is quite clear that it is a relatively unlikely event in relation to what the phenomenon of protein divergence would be, which is how we think most proteins would have evolved, which we see in this particular tree. So let's see, what are we seeing in particular? It is a maximum parsimony tree that was built from a presence-abscessence matrix, evaluated from 776 folds in 185 genomes from practically the 3 domains of life. Understand what would be archaea, bacteria or eukarya. And then we can design metrics that help us estimate precisely the diversification that occurred in each of these lineages. So this is what we are seeing in this gray bar, which represents precisely the regions where considerable diversity has been detected by some combination of these lineages, such as it could be only the contribution made by bacteria, only the contribution made by archaea, or the contribution of what would be both archaea, bacteria, and eukaryote represented in this black line. And so now, thanks to this tree of maximum parsimony, we can build a tree that would approximate what the evolution of how proteins occurred would be, where we see that there are two main phenomena, where what we find is first a diversification at the architectural level, where there is a contribution, quite considerable on the part of the three domains, as well as what would be the bacteria and the eukaryotes, and that subsequently a diversification phase occurs at the organism level associated more with what would be each of these specific domains, as we can see in these regions, where there is obviously a greater contribution either at the level of bacteria. Surprisingly, the archaea have a contribution that could be relatively small in relation to the others, which would be the combination of both archaea and bacteria, where proteins are now diversifying, but within each of these domains. So what the image suggests here is that precisely before this entire process of organismic diversification occurred, a diversification process at the level of protein folding also had to occur. And when we look at what kind of proteins are found in the most basal nodes of our tree, we also found some of the faults that we think have been the oldest based on multiple analyses. And among them, curiously, we find precisely this type of folding such as the P-loop, which, as we previously reviewed, is capable of binding to phospholigands such as ATP, and that even this activity of binding to phospholigands, as we reviewed, evolved at least 275 times independently throughout evolution. Later, we found other quite successful folds that can even be considered the superfolds that we also previously reviewed. Among those, we find what would be the thin barrel, a quite successful fold in terms of the functions that we can find that can be performed thanks to this specific folding. The Rotman folds that we also previously reviewed, and that, as we saw, up to 20% of all the proteins present in the PDB. They share what would be the minimum Rothman structural motif, among others, foldings such as ferredoxin and flavidoxin, example that we also already investigated in the class of structure of proteins, among others, and here then the idea comes from this opinion article discussion, which I highly recommend you review because it really covers well what the thought would be evolutionary in this sense, which is called evolution and tinkering, is a word that does not have a direct equivalent in what would be Spanish. We could even think about it in evolutionary terms. That is, it does not have a Spanish counterpart. In such a way that tinkering means this creative process where you have elements available at hand, and with them, you try to solve the task that is presented to you at the moment. So let's say that the perhaps most appropriate translation would be as evolution. An improvisation, although improvisation is not a word that recapitulates all the information that the word tinkering seeks to reflect. And so, in this comment by Francois Jacob, a lot of this evolutionary thinking is really covered, whether at the organic level or at the biomolecular level, where what is really suggested is that evolution operates on the pre-existing elements in the system, and it is thanks to this creative process where multiple forms can be reconfigured, something that we can see precisely reflected in what would be this first stage of structural diversification of proteins, and the subsequent diversification at the organism level, in such a way that really the elements that make vertebrates or practically all species different from each other is not really the ingredients that make them up, but the way in which these components are regulated. And in addition to this, it would evidently be the history associated, the regulation that led to precisely establishing said evolutionary lineage. When we talk in terms of protein regulation, as we previously reviewed in the protein function class, there are really multiple levels at which all protein activity can be regulated, whether at the genomic, transcriptomic, proteomic, or even phenotypic level, finding precisely this histone code that we already explored previously. Other types of post-translational modifications, such as phosphorylation, which so quickly our ribosome translates its sequences into proteins among multiple other characteristics, such as we can point out, would be the subcellular location, where different biochemical environments occur, such that then now the folding would adopt a different behavior than that it had in another subcellular compartment, depending specifically on the tissue where it is located, among multiple other aspects that, as they observe, a quite exquisite regulation occurs so that the needs of the organism can be satisfied according to its environmental conditions. So we return to what was our basic scheme of what the evolution of proteins is, and we have already reviewed what probiotic chemistry was. We have already reviewed the primordial peptides, and so now we proceed to review what would be the first proteins on this primitive Earth. The idea is that thanks to these small structural segments, these fragments, motifs, or structural signatures, whatever you want to call them, it was possible to create highly symmetrical forms, as you can see in these structures, where we see that these small motifs can be duplicated relatively easily, and subsequently we can proceed to what would be their diversification in structural terms. Here what we see is that, for example, the protein to the left, a team barrel, we can divide it into two structural motifs, or for example, this protein signaled here by the indicator. We see that it may have several copies in this sense. And here one relevant thing to observe is that it is thought that precisely in the nooks, or in the most central parts of these duplications of these structural motifs, is where this capacity for different ligands to bind to it could also have evolved, such as metal planes, osciligands, and even interactions with other proteins. And even if we take precisely this protein called beta-propel, and we predict the probability of each of its residues that they can bind to ligands, it turns out that precisely these residues that are relatively isolated from what would be the solvent with respect to the rest of the protein could precisely serve with a greater probability to bind to some type of ligand in particular. And that is precisely what we now cover here, scenarios in which proteins also evolved now also in relation to the molecules with which they joined, in such a way that the idea of conformational selection is suggested. As we reviewed previously, proteins are not really represented well, so it is a static idea of the protein, not even in its dynamic form through molecular dynamics assays, but really the protein as a whole. It is this conformational assembly where we practically start from the unfolded peptide to its native structure and its consequent structural diversity in what would be its specific environment within the cell or perhaps even outside the cell. And then, thanks to the understanding of this conformational assembly of proteins that would represent a particular fold, we can explain what its interactions with a ligand would be. That some group of sequences, due to the three-dimensional arrangement of their residues as well as their biochemical identity, since they were favored precisely by the interaction with this type of substrates, such that then now this union could be established that what it did is that some of these conformational states within this energy landscape were perhaps selected for their adaptive value by being able to bind to this type of substrates, and that subsequently, thanks to the establishment of these contacts as well as other mutations, that would help to stabilize what would be the three-dimensional folding, given what would be the modification to the substrate, since these relationships between what would be the proteins became more and more sophisticated with several of their dealings, with which they can interact. And here something relevant again to consider is the contribution that precisely the metal ions had during the first stages of life. Well, as we previously reviewed, what would be the coordination geometry provided by this type of metal ions is quite characteristic and also provides part of what would be the geometric basis, the structural basis, for proteins to establish their three-dimensional folding patterns. As you can see, what are the iron and nickel atoms, since they precisely have an octahedral geometry? Likewise, other types of metal ions also have their own characteristic geometries. Which they do is reconfigure this geometric environment on which proteins must attach in order to interact with this type of ions. Then we review what this diversification of structures is based on their elongation. In this case, what we do is clearly review, such as the case that would be associated with what is known as the hub domain, where we see that this would be like the central part of this domain, which is made up of 5 beta and 4 alpha helices, which would really be like a relatively small structural motif. However, what we see is that the hub domain, since its members in several superfamilies, are actually now found in a more extensive way in what would be mainly its end associated with the terminal carbon in such a way that this part then elongates, and precisely more loops begin to appear that what they do is a reflection of how this specific type of folding is gaining more. And when we look at the different types of superfamilies that are associated with this folding, we observe that all of them share precisely this central region. But now there are others that are considerably more decorated with some other type of secondary structures, and this is also reflected by the number of residues they may have. In this sense, for example, this protein called Higley-1 has 323 residues compared to what would be the central idea of the hub domain, which only has approximately 130 residues. Then we see how proteins are also diversifying in terms of the mass. They can contain and evidently adding new elements of secondary structures that may or may not be related to what their function or structural stability within the cell would be. Yet another idea is that foldings can even be recombined. As we can see here, we have 2 types of folding, one marked in green and another marked in blue. And the idea is that through phenomena such as gene fusion, duplication, or splicing, precisely these types of folds at the level of the genes that encode them are now reflected in what would be the fusion of these 2 types of domains in what would now be a single protein. And obviously, there are multiple mechanisms that can create what would be the diversification of proteins, such as gene fusion, the extension of the exons, recombination of the exons, and as you can see, practically what this contribution from the alternative splicing machinery would be like a great center of diversification for proteins in this sense, hand in hand with what would be its phenomenon associated with what is the elongation of proteins, as we reviewed with the hub domain, in such a way that we can also conceive what is known as a phenomenon of creative destruction, which is a concept that usually applies to various areas, for example in economics, in such a way that what we have is the idea of an ancestral folding, and that due to these phenomena such as duplication or fusion, then now we have precisely an arrangement of 2 ancestral folds that in this case are fused through what would be the interaction of their spin regions or their loops. So this also reconfigures a whole possible combinatorics of interactions that there may be between these 2 foldings in such a way that this interaction then matures until it achieves a certain adaptive value for the circumstances where it is located, and then in this adaptive process we move from the idea that these 2 foldings that were joined at that time are completely modified what we could associate with what would be their 3-dimensional folding patterns, such that now we have a result that would be like the daughter folding, which preserves parts of its 2 parent folds, but is clearly completely different from each of them. And perhaps this explains why we find these structural signatures in different folds that apparently have no homological relationship, but are precisely present in multiple folds. In order to further review how this process of diversification and evolution of protein architectures occurred, you can review this review for redundancy, which is quite nice in my personal perspective, which is precisely called the natural history of protein domains, and which we can really also bring to the table as a kind of analogies too. Understand more how diversification occurs at the protein level. For example, making an analogy such as the endosymbiosis event, which we tend to understand more easily, such as this idea that what would be the mitochondria was actually another independent organism that throughout evolution established interactions with other types of cells, such that it was now integrated within it, and now functions practically as an energy center for most life forms. And then this phenomenon of endosymbiosis could be reflected as this idea of being able to recombine foldings, and that now from the phenomena of duplication, recombination, and in general alternative splicing, we have precisely new foldings. Another idea that would also have an analog would be this idea of what horizontal gene transfer is. As you know, the bacteria present in a particular environment can share their genetic information through plasmids, and thanks to this interaction with plasmids, they can obviously gain some activity that they did not previously have, and that, thanks to this plasmid, they can carry out. And then perhaps an analogy equivalent to what the world of proteins would be to what this horizontal gene transfer is would be. It would be the presence of these structural motifs that are present in a number of foldings that apparently have no evolutionary relationship, but that have precisely this characteristic structural signature that may be advantageous to them in terms of their folding over time. So okay, now we're going to talk about the sequence space, which, as I mentioned, it is a key concept that helps us understand how all protein biodiversity was generated, thanks to the effect of mutations. As we can see here, there is an enormous diversity of proteins, and it is quite interesting to think about how evolution found the way to generate this enormous biodiversity of forms, of chemical behaviors, among multiple other aspects that proteins represent. This concept was originally proposed by a great scientist called John Maynard Smith, through what are like a kind of letters of responses to various discussions that were published in Nature, where he suggests that we can think precisely about the evolution of proteins through this concept of sequence space, such that then what we can think about is proteins in the form of a continuous network that is obviously interconnected to each of its elements. Each of its nodes would represent a protein. And then evolution can go through this network through mutational steps that do not necessarily have to go through non-functional proteins, but precisely through proteins that have a certain adaptive value. Even within this comment that John Maynard makes, he has one of my most favorite phrases in the world of biology, which is that we can also think of proteins and their corresponding genes like a series of isolated islands in a sea of sequences and meaning. A somewhat poetic version of thinking about what genes would be in that sense. So let's see what this idea of mutational steps through intermediaries that are actually functional means. For example, we can think that there is a probability of moving from the word to the word gene. As you can see, it has 4 positions. And that in combinatorics, at least in the English language, well, we have 27 elements, and I don't remember what the letters are that make up this language. So, what we can review is what set of word combinations can take us from what would be the word to the word gene. We can review in this suggestion all the way down that we can use what is the word, then year, and gene. In this case, we can find that these words may not have no sense, so in linguistic terms we could think that they have no adaptive value. Contrary to what the words would be in this other suggestion, we do not have the words worth, or go, gone, and gene. In this sense, we do find an interpretation of why they are words. Let's see them as adaptive, by which we can precisely say, cross this sea of words, and we could make an analogy with proteins, because now we could cross this sea of proteins through mutations that do not affect the stability or functionality of our protein. And then precisely here 2 key concepts come in. One which is precisely the adaptive mutations that would be mutations that are managed to be fixed through natural selection, that confer an advantage in what is known as fitness. A relative metric that we now move on to review is fitness. Fitness, as I mentioned, is this relative metric that means that a variant of a protein may be able to survive, function, and increase its frequency in the population in such a way that we then have a whole possible combination of proteins, represented by what would be this two-dimensional map. And on the y-axis, what would be its adaptive value within the cell for a specific environmental context, such that we can then traverse this sea of protein biodiversity, say from a variant protein 1 to a variant protein 2, through multiple steps of adaptive mutations to reach this peak that now in relation to this other protein. It offers greater fitness that may be clearly advantageous to the organism that has this specific variant within its environment. Do this, it is also evidently necessary to understand what the effect of mutations at the DNA level would be on their respective translated sequences. In general, we have two main types of mutations, those that would be mutations to a single specific residue, or mutations that are associated either with the insertion or what would be the removal of one of the residues. When we see this type of insertions or injuries, completely catastrophic changes can really be generated, although perhaps not necessarily, where we also have to refer to this combination of insertions and injuries with the word indel. In the sense when doing an indel, well, we can completely change how the nucleotide triplets are read, that is, a codon, in such a way that what would then be the sequence of our protein when a nucleotide is introduced or removed. Well, now we completely change this reading frame. And then, precisely, these insertions can make a catastrophic change at the level of the protein that it can encode, given that the codon is now changed, and perhaps a terminus codon could be inserted or a codon that is from an amino acid whose chemical behavior is completely different from what was previously present. And when we talk about point mutations, we also have different subcategories of the type of point mutations that exist. For example, there are some mutations that are considered synonymous because, as we previously observed in the standard genetic code, various amino acids can be encoded by different numbers of codons. In this case, if a codon is used that codes for the same amino acid in terms of the protein, perhaps it translates precisely to the same residue, but perhaps this property that is not affected at the protein level is not necessarily translated. It is transferred to what would be the replication or translation process. We also have mutations that are not synonymous because then, now the identity of the amino acid is being changed, and we even have mutations that are considered meaningless because then a terminus codon is included, and then what would be the protein is now completely truncated, and this is precisely the value that we can associate with each of these mutations. So we can talk about a mutation being neutral, when it really does not have a significant impact on the fitness of the organism that is providing this protein variant. However, we must also consider that precisely these neutral mutations are not necessarily neutral on these two other scales that I mentioned was the replication process or protein translation, because, for example, how the polymerase is coupled to different codons also has an effect that could perhaps promote or decrease its expression at the cell level. We also have mutations that, evidently, due to their particular arrangement of amino acids or the context in which they occur, may carry an adaptive value for the species that provides said variant. Therefore, they would be considered beneficial mutations. And deleterious mutations, since they would obviously be those that are already made, cannot be tolerated by the physiology of the organism in its specific environment. Or, well, completely lethal because by truncating the protein, you simply do not have that resource now to, for example, synthesize proteins because it turns out that it is a ribosomal protein, and then now you are left without a relevant part of your ribosome, and therefore you affect everything that would be its translation machinery, for example, This idea of beneficial, deleterious, and neutral mutations is also partially reflected in what would be the idea of the degeneration of the genetic code, which precisely, as we reviewed previously, there are some residues that precisely have a greater number of codons. And we think that what would be the first residues, these hydrophobic residues, as we can see in this diagram. Precisely, they had more codons, and what would be the most recent forms of proteinogenic amino acids now have a smaller number of codons, which is precisely what we currently see. For example, what would be the last residues that could have been added to this alphabet of 20 amino acids would correspond to what is methionine and tryptophan, and it turns out that in what is the standard genetic code, since these amino acids simply have a simple codon, in relation to what would be, for example, these first amino acids such as glycine, lannin, which have 4 codons that code for them, and therefore, if any change occurs in this sense, what would be the translation of this specific codon is not so affected. And we even see some residues, such as serine, arginine, glycine, which very interestingly have 6 associated codons that can code for said amino acid. This is precisely the degeneration of the genetic code, such that it is then generated as a buffering effect, such that this genetic code precisely allows for buffering genetic errors, but at the same time also allows for the evolvability of our biological systems, an aspect that we will review later. It is also important to consider how we can precisely use this evolutionary information in our algorithms, as could be represented by these matrices that we already reviewed, which are the blossom substitution matrices, which precisely what they do is reflect this evolutionary probability that amino acids can occur through what would be the empirical observation of a set of diverse sequences. As we can see, it is not the same to change from one cysteine to another cysteine under the biochemical context in which it occurs, just as it would be the change from one tryptophan to another tryptophan, or completely changing the biochemical identity of what would be the side chains, which in this case would be penalized with a negative value within what would be this substitution matrix. The idea is that we then have to consider this phenomenon of neutrality, such that proteins can then traverse this sequence space through what is known as neutral drifts, where precisely these regions are avoided, these configurations of amino acid sequences that do not provide an adaptive value, and therefore their fitness would be relatively lower than any specific variant. So again, we went from a protein variant number 1 to protein variant number 2. Along mutations that now do not necessarily have to have an adaptive value, but it is simply enough that a neutral value is present, so that it does not considerably affect its function or its structure. And then we avoid precisely these valleys of fitness, where perhaps the mutations are now meaningless or are completely deleterious, and then there simply can no longer be evolution in this sense. To this evidently this landscape, this sequence space where proteins can traverse in a neutral way, it is relevant to consider that it evidently corresponds to the effect of 2 major elements. One that is precisely the environment and the selection pressures that it provides, as well as what would be the genetic context that originates from said organism that has said variant. So really, what is this sequence? This idea of neutral drift must be understood from the hand of these 2 branches, these 2 concepts, such that then now it is actually more correct to say that a mutation is really conditionally neutral with respect to the conditions of its environment, and precisely the environment or the genetic context where it occurs, since there may even be several systems such as the chaperones that could assist the folding process in the event that perhaps the mutation has a deficiency in terms of its folding, and that then thanks to this type of protostatic mechanisms, then the cell can address precisely these problems, although perhaps then this variant does provide some adaptive value, but that during its folding process, this mutation, well, perhaps it is not so advantageous, but thanks precisely to the chaperones, now we can contend with that Effect right. As you can see, precisely what evolves is the whole, although usually we only indicate the biological object that we are trying or pretending to explain. And here another key idea is also to understand that precisely these neutral mutations what facilitate is the evolvability of the system. This capacity precisely for evolution to evolve, something that is a relatively complicated concept when it comes to discussing, and that is done between colleagues who are from the world of evolutionary biology. And it is even more complicated when it comes to areas outside of what is evolutionary biology, as would be our case, as we are more on the side of protein engineering. So then it is relevant to know what is known as evolvability, which is precisely this ability to be able to adopt new functions from precisely these existing faults, even form completely new folds. As you can see here, represented by chems, where this evolvability led to a diversification of each of its behaviors in specifics, which in this case, in terms of proteins, would be this ability to adapt, even considering few changes at the sequence level. And the concept of evolvability also goes very hand in hand with another one, which is robustness, which is precisely this idea of tolerance at the level of either the structure of the protein, at the level of the function of the protein, with respect to mutations, in such a way that precisely the desired phenotypes are conferred in that sense, because there will be some proteins that, due to their specific architecture, well, perhaps they are more delicate with respect to whether you mutate an amino acid, with respect to other quite successful foldings such as the team barrel, than perhaps then in relation to this other hypothetical protein, since it may be able to tolerate a greater number of mutations in thermodynamic terms of the structure of the protein, as well as in terms of the functions that can be associated with each of these residues. So that then it is precisely both robustness and evolvability, two concepts that are conditional with respect to what would be the selection pressure that the environment provides to the specific cell. Thanks to these two concepts, we can also understand this idea of what fitness landscapes are, in such a way that not all proteins share this specific evolutionary landscape, right? There will be some proteins that can generate phenotypes that are not as variable. They are relatively easier to be explored in evolutionary terms, such that simply the desired or minimum phenotype necessary to satisfy their functions within the cell is relatively easy to achieve, represented by a single peak, where multiple variants can actually navigate through this evolutionary landscape in a relatively easy way. Contrary to what would now be evolutionary landscapes or fitness landscapes, whatever you want to call them, which are now relatively more complicated because here, what would be the adaptive value of all these variants would be equivalent. However, being able to traverse throughout this entire evolutionary landscape is now much more complicated given that we now have a multitude of peaks where it is now evidently more difficult to be able to advance in terms of the evolutionary trajectories that can be associated with each of these states, and even as we have other types of configurations of these adaptive spaces, where, well, now we already have quite unequal peaks, as you can see here, the vast majority once again reaches a state of fitness, where the variants are advantageous for the organism that has them, and which in this case cannot necessarily reach precisely this maximum associated where there is only one evolutionary path where we can precisely reach this state, but the rest is now diminished in relation to what this maximum peak would be. So in this sense, the evolutionary landscape is even more complicated to be navigable. Within this idea of evolutionary landscapes, it is also relevant to consider two major aspects. One that is precisely the importance of the thermodynamic stability that proteins need to adapt their three-dimensional folding, and subsequently see what function they do. As you remember, precisely what we can do is verify that our protein folds well, and later we can ask it what things you are capable of doing. But if you are not able to bend, it is more likely that you will not be able to carry out your functions. Except for what would be these intrinsically disordered proteins, as we previously reviewed. Although in general terms we can start from this premise that first it is necessary to bend yourself, and then we ask you what things you are capable of doing. And another concept is the idea of promiscuity at the level of function, something that we are going to explain through this diagram, where what we see is this sequence space represented by this little map, where each of these points is a variant, and each of these colors is a superfamily within what would be a particular folding. And what is on the y-axis, we see what stability is in terms of thermodynamics. Therefore, then what we see is that the sequences that have a great thermodynamic stability are really usually associated with the presence of what are mutations that have a more neutral behavior, and therefore precisely. It also makes them more robust to the effect of mutations. And so, thanks to these robustness, we can now explore different capabilities of being able to perform functions that are relatively similar in terms of the biochemical reaction that occurs. For example, we can schematically associate each of these superfamilies in this specific figure with some function. But they all have a certain similarity in terms of the reaction. Therefore, those sequences that, for example, could be associated in this cyan functional space will perform only this specific function. But those protein variants within this superfamily, we now see that they overlap with two other superfamilies, which would be, for example, this one in red and this one in royal blue. So that then, the protein variants found in this region, now they are not as efficient in carrying out this function in cyan, but now they are capable of carrying out these three functions to a greater or lesser extent. And this is precisely what is known as promiscuity. The ability to carry out related biochemical reactions, and that is not necessarily its best performance in this sense. And precisely thanks to this promiscuity, multiple protein variants can be explored, which are always based on thermodynamic stability, so that the variants that do not precisely have great thermodynamic stability perhaps are reduced in their exploratory capabilities in terms of sequence space. Again, the idea here is that we can precisely associate what evolvability is as a kind of starting points that are seen, conditioned precisely by this property of thermostability, and that, as we reviewed previously, proteins are actually marginally stable given that we really want proteins that are highly dynamic and that are not completely a stone, but we also do not want proteins that are completely disordered and that do not adopt any three-dimensional configuration in space. Something that we can see through this scheme reflected in what would be the level of activity of some specific function in symmetry, as well as the multiple starting points in which we can advance along this evolutionary landscape, such that there will then be a level associated with fitness that results from a physiological relevance. And then this level of activity, not having been satisfied by the particular configuration of amino acids that constitute this protein, well, this threshold that is physiologically relevant is not reached, and then it is left out of what the exploration of this evolutionary landscape would be. We can also see it here, now in terms of what thermodynamic stability would be and not necessarily fitness, that in this case we see precisely different evolutionary trajectories, whereby not having sufficient thermodynamic stability, we once again reach this margin, where there is no longer really a physiological relevance for the cell. And then this combination of residues is discarded, and we now start from this other example where this protein can precisely operate within this margin, which is precisely this marginal stability, and which can even contend with what would be mutations that could deteriorate its thermodynamic stability, and that thanks precisely to these compensatory mechanisms such as the chaperone system. Well, its abilities to be well folded inside the cell, to be able to carry out its functions are now rescued, and thus it can navigate along this evolutionary landscape, along either neutral mutations or hand in hand with this type of compensatory mechanisms. Again, we must obviously think that adaptive mutations, even and in a very curious way, can present destabilizing effects more recurrently than we would like. So it is thanks to this type of compensatory mechanisms that these types of beneficial mutations can be considered beneficial despite their effect, which is destabilizing. Thanks to these other mechanisms, we can now navigate more easily along these evolutionary landscapes, and then we also have to consider in terms of what the thermodynamics of the protein would be. What effects would it have, for example, if mutations occurred at the level of the core of hydrophobic residues that were buried, or at the level of the residues that are exposed? So that, for example, what would be mutations in the surface of the protein can cause changes of around 0.6 kilocalories per mole. Always remembering that proteins tend to be in a range from fully unfolded to external native structure of approximately 5 kilocalories per mole. In that sense, when we now mutate residues that are precisely associated with this hydrophobic core, with this hydrophobic center that is evidently an essential part during the folding of the protein, we now find that the changes that are usually associated with this type of buried residues reach approximately 1.4 kilocalories per mole. Therefore, it is more likely precisely that mutations could occur in what would be the surface of the protein with respect to the center, because in thermodynamic terms, their effect is much stronger, as we already observed. To better understand this relationship between functional promiscuity and protein diversification, let's review this example within what is known as the PVP superfamily, which stands for penicillin-bending proteins. In particular, here what we are seeing is a network composed of approximately 600,000 sequences, where we see different lineages that have diverged from three main groups of PVP proteins. Some are called low molecular weight PVPs, and others are called high molecular weights, that is, low molecular weight PVPs or high molecular weight PVPs. We can observe that these PVPs are indeed associated with what would usually be their PVP activity. However, we observe that there is a path along this evolutionary trajectory that now takes us to another group of enzymes related to these sequences that are now capable of carrying out a beta-lactamase type activity, such as class A, class C, or even class D beta-lactamases. And we see that in turn, this superfamily is also related to other types of proteins that have some other type of functions, such as esterases. And the idea here is that precisely through this functional promiscuity, the sequences can diverge and obviously diversify both in terms of the amino acid sequence and in terms of the structure. Something that we can review when we compare the structures of a class A beta-lactamase in orange and a PBPA, which would be their last common ancestor, which is indicated in blue. When we align the structures of these two proteins, we observe that there is a large overlap in this sense. There will be some regions that seem to be characteristic, indicated as a little more transparent in color, but in general terms really, well, almost all of the structures are quite similar. And when we look at the metrics related to structural similarity, such as the RMSD or the GM score, they really suggest to us, due to their values of 2.22 or 0.87, that it is really the same folding and that they even share approximately 21% sequence similarity. However, when we now compare a class A beta-lactamase in relation to what would be a low molecular weight PPP, now we notice greater differences in structural terms. In the previous case, perhaps that structural similarity is not framed, and then it is necessary to turn to the similarities that we can find at the level of what the sequence of residues is. And in this specific case, we can see in a quite noticeable way that it has the presence of regions of folded veins that are not present in what would be the class A beta-lactamase, and that even when we turn to see what their structural similarity metrics are, it suggests a lower degree of similarity in relation to what would be the PVPA, which in this case could have 3.12 in RMCD values and 0.79 in TM score values. It is then suggested that it is the same folding, but you can see that there are structural differences that are relatively considerable, at least within the term of this superfamily, and that are evidently reflected in their functional capacities. In fact, when we review what the structures associated with each of the types of beta-lactamases are, we observe that even at the sequence level there are conserved residues that are catalytic, and that are therefore relevant to be able to perform their function. As we can see here, what would be Class A, Class C, and Class D of thalactomases have at least 3 motifs at the sequence level, and the vast majority present this combination where there is always a serine spaced by 2 residues followed by a lysine. In the second motif, we usually present what would be a serine followed by an asparagine, but then we can find some variability in terms of which serine would now be replaced by some other amino acid such as a tyrosine in this case, and so each one obviously found a certain adaptive value in each of these residues to be able to carry out the activity of beta-lactamases, which is even reflected at the level of their specific microenvironment related to what would be the active site, as we can see here in such a way that in each of these active sites, well, it is related to what would be certain loops that are precisely surrounding the active site, and that would serve as a gate to access this active site. And it is thanks to these loops that we can regulate what would be the effectiveness of this catalytic activity, which is to degrade beta-lactam compounds, which even so when we talk about this type of compounds, we are really talking about a great diversity of molecular structures such as could be represented by beta-lactam antibiotics such as penicillins, carbapenems, monobactams, or cephalosporins, of which they are further diversified within each of these specific groups. So here the question that is natural to ask is why is it that there are some folds that have hundreds of enzymatic functions, while there are other folds that only have a few? Here we can observe 2 large superfamilies. In this case, it is a superfamily called 310450, and in this case, it is another completely different fold, 3.202 and blah, 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 where we usually associate these IDs of what the CAD system is with what would be the superfamily associated with team barrel, and in this case, it would be the superfamily associated with this nuclear transporter. And as we can see, when we compare the number of enzymatic commission numbers, these numbers called S terms, we find that in what is the Tim barrel superfamily, there are 160 possible enzymatic activities associated with this folding, well, specifically with this superfamily. And when we review how many enzymatic activities are now usually associated with the nuclear transporter superfamily, there are only 24 terms in this sense. And as you can see, it is mostly of the oxidoreductase type. It is represented here in blue, while in the timbaran superfamily there are transferases, there are lyases, there are isomerases, among other types of enzymatic functions. So it is quite clear that there are some foldings that, perhaps due to their 3-dimensional, thermostable properties, among other factors, have more functions in this sense. And then here arises a term that can help us understand why this type of phenomenon occurs, which is called polarity, which precisely refers to a certain modularity that exists between the residues of the active site in relation to the residues that constitute the backbone or scaffold of the rest of the protein, in such a way that these 2 groups are relatively separated from each other, so that they allow a certain degree of freedom throughout evolution. For example, we can talk about the fact that this folding that is a glucocorticoid has a low polarity because, approximately 90% of the residues that make contact with its ligand actually form part of what is the scaffolds of the protein, its body in this center. Here we see in color exactly the ligand, and what would be in orange are those residues that establish contacts 4 Armstrongs away. As we can see, there really is a considerable amount of waste. When we compare it with now another folding, which would now have a medium or medium-low polarity, in this case again represented by a class A beta-lactamase, where we can observe that now the number of residues that are establishing contacts with what is the ligand is smaller, and that actually certain ones are even seen in its surrounding environment. Loops that could precisely modulate their catalytic activity in an easier way. So here it is clearer precisely that since these two types of residues are separated, those destined a little more to their catalytic activity compared to those others destined more to what would be their structural stability, since there is a greater degree of freedom, and therefore there would not be some great coevolutionary forces that, well, limited in terms of divergence at the sequence level and at the function level, obviously, for example. Then here we see precisely these loops that are in an area surrounding what is the active site, and these loops obviously have a certain flexibility like any other loop. In fact, as we reviewed in what the class of protein function is, we found that precisely this degree of dynamism, represented here by this blue club and this other green one, where it is the blue that is more dynamic, since they really represent what would be a kind of personality of each of the folds, such that then it is as if there were a specific fingerprint that gives it a certain behavior. It is in this way that the folds that have a low polarity, then, they find a certain coevolution between what these types of residues are from the active site with respect to those from the scaffold, and therefore the divergence is more limited. And otherwise, those folds that have a high polarity, then the catalytic residues can diverge in a relatively easier way, which allows the exploration of functions that are associated with their neighborhood of this functional promiscuity. Obviously, also to be able to conserve what the function of a protein would be in its specific biological context, it is because this protein is providing a certain adaptive value. Otherwise, then it is energetically costly to maintain the protein, something that we can address with respect to what would be the divergence or evolution of protein functions. For example, we find that an enzyme will precisely maintain its hematic functions, but it can even change either the substrate or the chemistry. In the event that you change the substrate, the same chemical reaction can be maintained, but in this case, obviously you change a substrate, such as carrying out a hydrolytic activity on a peptide bond, and eventually adapting to another type of bond. Otherwise, for example, now in what would be the change in chemistry, now we find that the substrate is preserved, but now this same catalytic action is being carried out through a different chain of biochemical reactions, which in this case could be represented by the idea that the identity of some catalytic residue is completely changed, and this same catalytic activity now has to be accessed through different catalytic mechanisms. This is the divergence that there may be within the enzymes but preserving their enzymatic function. However, it turns out that there are some proteins that operate in a range where they preserve their original catalytic activity but have also now unlocked, let's call it another capacity, that is not directly related to their enzymatic capacity, such as serving as a transcription factor, structural relevance, among other aspects. And in this case, we call this type of protein moonlighting, as it could be in the case of enolase, a protein that, as its name suggests, has glycolytic activity, but also now has a role that is relevant in structural terms of the cell membrane. in the event that these functions have been relatively well preserved. We can find precisely these two types of activities in this type of enzymes called mulating. Thus, we can also find other types of enzymes, which we call pseudoenzymes, which actually have a greater tendency to have completely lost their catalytic activity and now completely fulfill another function within the cell that is not associated at all with their enzymatic activity from previous ancestors. As in the case of pseudoproteaceae, that, as its name suggests, precisely its previous lineages, its last ancestors perhaps could have had precisely this protease activity, but in this case it was completely lost and now fulfills some other function as a trigger of signals, among other types of functions that any protein can occupy within a cell. And the idea here is that precisely being able to maintain what is the enzymatic function really represents a cost for the cell. It is not free to express this type of proteins. For example, we can find that when we have plasmids that express genes related to antibiotic resistance, such as beta-lactamase enzymes, it really tends to reduce what would be the fitness of the generations of bacteria by up to approximately 3%. And this is because now you have to allocate resources that the cell has for what would be the constant expression of this type of enzymes to be able to deal with media with the presence of antibiotics. Therefore, you have to redirect aspects such as the amount of ATP you have, the amount of amino acids you have available, and all of them obviously to be able to create this type of enzymes. Even what is the energetic cost of different molecular entities is relatively registered. For example, there will be some molecules, such as carmagulin, that have an energy cost of approximately 4.3 times 10 to 3 molecules of ATP, which could be a relatively cheap cost in terms of a protein that is 148 residues long. However, there will be other molecular components that are relatively more expensive in energetic terms, such as the ribosome. In this case, the ribosome costs in terms of ATP approximately 4.3 times 10 to 5 ATP molecules. And so we can associate an energy cost to each type of subcellular region or membranous compartment of what makes up the cell, in such a way that really resources must be allocated to the most essential elements for the cell to survive. If in this case we need to express an enzyme that allows us to survive in an environment that precisely has antibiotics, then there is a certain adaptive value in allocating resources towards this type of mechanism. Otherwise, then the cell will tend not to express this type of resistant enzymes, and then allocate them to other sections such as the ribosome. So now we move on to what is the third part of this class, where we evaluate the effect of interactions between proteins, as well as between other molecular entities, a phenomenon we know as epistasis. The idea is that, again, by being involved in a multitude of interactions, proteins actually establish relationships between residues of the same chain, as well as the chains of other proteins, in such a way that we can find these evolutionary couplings that what they really reflect is a coevolution between these types of residues, either within the same protein or with other proteins. And then, if an amino acid mutates, then the corresponding amino acids that establish the interaction also have to mutate in order to satisfy the requirements of said interaction. Here we can see different types of interactions, such as interactions of residues within the same protein, interactions in a multimer such as different chains, where in order to establish a certain stability, they have to interact, or even interactions between what the protein is and its ligand that can even be relatively relevant when we find that also what it is to address precisely the ligand to the protein. There may even be large conformational changes, which are stabilized due to the interactions of residues that are apparently distal. But when you look at the substrate, they turn out to be relatively contiguous. Or even when we talk about metabolic networks, such as receptors associated with G proteins, there is evidently a whole cascade of interaction where we can also see a certain degree of coevolution between residues reflected. And even this concept of coevolution was precisely exploited, in informational terms, to be able to obtain evolutionary information of said residues that co-evolved in order to predict the structure of proteins using the AlphaFood algorithm, an aspect that we precisely review in its particular class. Then we understand well what the concept of epistasis is, which is a kind of nonlinearity between the effects that can be associated. To each of the mutations, then, let's review these two examples of two fluorescent proteins. One blooms in blue and is called MTAG, and the other blooms in red and is called MGATA. The idea is that these two proteins are derived from a common protein, and that really the only thing that distinguishes them is a total of 13 mutations, that if we see it in terms of combinatorics, we would have approximately a number of 8192 protein variants that we can combine using. For example, position number 1 has 20 possible combinations, and so then with position 2, such that we can then consider this neighborhood and see what happens if we change one amino acid for another, and if we can perhaps follow a trajectory from a green color to a reddish color. And this is precisely what we observe. For example, if we start from a reference protein that in this case has a phenotype that is usually associated with a precisely blue color, we see that under this context of mutations that occur, precisely this blue color is presented. And when there is a mutation of what is a tyrosine for an arginine in position 197, this tyrosine coloration is now diminished in its capabilities, now represented by a black color. And as you can see, a relative score that indicates that it precisely lost its capabilities to flower in blue now. Let's evaluate precisely this same context of mutations now together with another set of mutations. In this case again, we start from this mutational context, and we observe that precisely through this mutation of a tyrosine for an arginine, its ability to flower in blue is reduced. However, it turns out that when we associate this combination of mutations together with a new mutation that would be the substitution of a phenylalanine for a serine, we now find a phenotype that has an expression that is already associated with a red phenotype. And that when we only evaluate what this mutation would be in a singular way, it turns out that its ability to express itself as a blue color is reduced, but that in combination with this other protein that first reduced it, now it turns out that its color changes completely, in this case going from being blue to red. And so we can consider the epistasis both firstly, secondly, or even thirdly, in such a way that we now have a combinatorics of possible mutations that do not necessarily have a linear effect on what the phenotype is. This is precisely one of the key concepts of what epistasis is, in such a way that precisely the impact of mutations, whether on the stability or activity of proteins, really depends on the entire previous mutational context where this mutation occurs, right? In such a way that this effect of the mutations is not really linear, but would really have a non-linear behavior, or if we want to see it this way, relatively contingent in this sense. Obviously, due to these interactions between residues of the same protein, as well as with what would be the atoms of a ligand or with residues of other chains. Well, there are a multitude of interactions that could establish precisely these phenomena that we evolve, as we can see in this specific case. And really here you can observe in more detail cases that we are not really going to address because they could be quite complex, that even epistatic dynamics are established. It is already done by dynamics of conformational selection. It would also be due to the interaction of residues, interaction with cofactors, among a multitude of other aspects that can establish this type of epistatic interactions, which we consider both a blessing and a curse. And this is because, if we consider that the effect of mutations is nonlinear, then it makes it relatively difficult for us to understand what our predictive capabilities would be in order to improve some enzymatic function that we are trying to design or improve. However, in the sense of a blessing, it turns out that now, on the other hand, we only need a handful of mutations that are strategic, that are in an appropriate context, so that a completely drastic change can occur in terms of its function or its thermostability. And it is quite clear that proteins are not really singular entities within the protein. Each protein actually establishes a multitude of interactions with the other proteins found within the cell, and which are really key in evolutionary terms. For example, in the case of a quite famous protein called p53, which is a transcription factor that usually regulates quite a few metabolic pathways related to a multitude of things. As we could see in this figure, we see that p53 is capable of regulating aspects such as the cell cycle, metabolism, apoptosis, and all that thanks to the proteins with which it interacts, and with which it has precisely co-evolved throughout evolutionary history. So you can notice that the evolutionary histories of each of the proteins are interrelated with the evolutionary histories of other partners with which they interact, either directly or even indirectly, which obviously makes it a contingent phenomenon in this sense. And when we now think about this contingent phenomenon in terms of the entire cell, it is quite clear to illustrate precisely this network of interactions through what are metabolic pathways. Here you can simply observe what would be approximately 750 reactions, and as a consequence also their products, which would be approximately 500 metabolites. And in each of these points, precisely one protein is also associated, with its functions also related to the promiscuity with which it can be related, in such a way that there really are no completely isolated modules within what metabolism is. Here, in fact, you can notice a certain margin of overlap between different modules, but really what would be, for example, a module here colored green in this region, since it would really be either directly or indirectly related to other modules within what is every metabolic system in this sense. To make it quite clear that the evolutionary histories of all proteins are actually related. And when we think in terms of epistasis, well, we could think about how far a mutation can have effects when we consider the diameter of a protein. And it turns out that in this case, again going to the favorite example addressed in this course, which is class A beta-lactamases, we find that what is a change in one of the residues, that is, a mutation within this protein, is reflected as variations in these convolutionary terms between residues of up to 20 Armstrongs in diameter, well, radius in the sense, right? That is to say, these frequencies that we can calculate through some mathematical models of what probabilities there are for a few pairs of residues or up to 3 pairs of residues to mutate. We see that they are modified when we make even a mutation that is approximately 10 Armstrongs away from said specific residue, which could be thought of as a kind of butterfly effect, where in this case, what is a mutation can have effects on other residues, even up to a little more than half the diameter of the protein, given that in this case this class A beta-lactamase has a distance of approximately 46 Armstrongs away, and its effects in terms of this evolutionary coupling between residues reaches up to 20 Armstrongs. That is approximately half its distance. Epistasis is also reflected as a limiting factor in terms of the reduction of possible evolutionary pathways that can be accessed through natural selection. a finding that is suggested from this particular publication, along with another set, where we can evaluate an example in which we see precisely a class, a mass-beta act, which is usually associated with the ability to catalyze what would be the hydrolysis of penicillins. And we found that, thanks to the presence of 5 specific mutations, its specificity for this type of substrates can now be modified to another type of beta-lactam antibiotics, which in this case are cephalosporins, more particularly acetaxmin. And thanks to the effect of these 5 mutations, the resistance against antibiotics that this enzyme provides to the bacteria can be increased by up to 100,000 times, which is considerable. So, when we talk about 5 mutations, and when calculating the combinatorics, we find approximately 120 possible evolutionary trajectories that we can obtain from the combination of said mutations. However, it turns out that, when we evaluate how likely each of them is to occur, well, only about 18 trajectories could be really accessible via Darwinian selection, since not all combinations can really offer this phenotype associated with an increase in resistance against beta-lactam antibiotics. For example, we can review the following evolutionary paths, which in this case only the 10 most probable are shown. Here we can observe what would be a first mutation of what is a glycine now replaced by a serine, and it is observed that this type of mutations can be assigned a greater probability, either because it has a contribution of greater thermostability, or because it can now precisely carry out what is more easily the hydrolysis of cephalosporins among other aspects. Subsequently, we now find a second mutation that perhaps in this case could once again contribute in terms of the catalytic activity of the enzyme, but which now has collateral damage that, for example, could be a reduction in the thermostability of the protein, given that perhaps now certain contacts that were previously there are no longer as established. And so we can trace different trajectories, such that we see that there are some trajectories marked here in red that are relatively unlikely in relation to others that seem to be much more favored. For example, we found that there is even a fairly well-known mutation when doing clinical research, specifically in hospitals when evaluating resistance against antibiotics, which is known as the global suppressor mutation, which in this case is this clinical mutation of a methionine for a threonine in what is position 182 of this beta-lactamase, such that it is quite curious that we always find this mutation, and that it turns out that its contribution in structural terms is to improve the thermostability of the protein. And so we can find different combinations of mutations that together would find what would be an evolutionary path, and that really due to epistasis. Well, it means that not all of them are precisely selectable because their interactions are truly nonlinear, as we previously reviewed. In fact, the evolution of proteins itself is related to multiple factors such as the level of gene expression, the mutational rate of what the protein would be, as well as its consequent recombination rate. Other aspects would be, for example, the selection of the cell that has said variant of the protein, that is, the dispensability of precisely that gene, which, as we saw, maintaining, for example, enzymes related to antibiotic resistance results in an investment of energy that could perhaps be used for other types of resources for the cell. And also now, what would be the interactions that these proteins can establish together with their partners, in terms of the fact that they are now precisely regulated directly or indirectly by these metabolic networks that are established. So that's how we can actually conceive of a network of interactions that reconfigure the abilities of a protein to evolve. In fact, when we think regarding the evolvability of proteins, we could make an analogy with what complex systems are, because both the evolution of proteins and this type of systems share certain analogies, in terms of which we already reviewed that include dynamics that are nonlinear. It is also sensitive to the initial conditions. Phenomena such as self-organization and emergence of new ingredients occur. Systems within the chaos that exists. For example, when we talk in terms precisely of this self-organization, since we find precisely the folding patterns that proteins adopt during the folding process, when we make an analogy of the concept of turbulence typical of complex systems, because we can speak precisely of the process of variation, in what this protein is in terms of its sequence throughout the evolutionary divergence, when we talk about this fractal geometry. Now we can make an analogy in how precisely they branch out through these networks of interactions that do not necessarily have to be non-functional, which is known as the space of sequences. When we talk about attractors again, in this case, it is very clearly reflected in what would be the presence of these superfolds, which turns out that for multiple evolutionary reasons dependent on these conditions of the primitive Earth, it turns out that they were favored in terms of the number of sequences that we can find associated with each of these foldings again, when we talk about what the sensitivity to the initial conditions is, because we talk now in terms of the contingency of epistasis, such that now it is really not so easy to be able to say that if we add two proteins that are beneficial, well, its result will be beneficial, but its interaction will be nonlinear. As well as with other concepts that do present a certain analogy between what complex systems are and what the evolution of proteins is, and which also invites us to think about whether the evolution of proteins is really something predictable, or even if evolution in general is something predictable. About which, well, it will depend a lot on who you ask, but let's say that here there is like a whole range of opinions that we can find on the matter. The option more to one side leans more towards the idea that mutations are completely random, which, as we already reviewed, or I hope I have been able to communicate those ideas, is really not entirely the case, because we as life, who are made up of atoms, could really think that mutations are not completely random, because we have to satisfy certain requirement imposed by physics. On the other side of the spectrum, we now find that what would be genomes, or what evolution is, it has a greater range, greater flexibility, in the sense of the laterality of the mutations in which it is associated. So even the other side of this spectrum, it could be interpreted in a somewhat more ridiculous way, such that mutations can be promoted by the power of the mind, which is also not something reliable, something not even thought of in these senses. So this would be like what the spectrum of mutation theory is, where perhaps we could think of ourselves as falling on this part more appropriately. In fact, there are also many discussions regarding this. For example, if the evolvability of a system is an evolvable attribute. Remembering that what evolvability is this ability to generate variation that is both heritable and adaptive, whether through genetic or non-genetic mechanisms, which really what they do is generate certain phenotypic variation, generate robustness to mutations, etc. Among many other things that, for example, we can see reflected in the same way in these other two publications, where we now also know that precisely one of the key factors to be able to give way to the evolvability of a system is the fact that it becomes thermostable in the case of proteins. But we also find certain biases, such as the fact that the vast majority of gene families in bacteria turn out to be really biased in terms of positions that they adopt within the chromosome, that even when we evaluate the architecture of the genome in terms of eukaryotes, we also find a quite specific regulation that we know as chromosomal territories, in such a way that there really are states of heterochromatin or chromatin that are unwrapped or enveloped to be able to express themselves, and that also when they are seen enveloped or unwrapped, they are slightly more exposed to being able to mutate in this sense. So now we come to what would be like a kind of a new integrative vision of how the evolution of proteins works, taking as a key what would be the recognition of the dynamics of the conformations that proteins remember are conformational assemblies that, although we usually see them as simple, relatively smooth valleys, they are actually quite rough because here there is a precisely promiscuity that interacts together with its ligands, and that evidently some of these conformational states are capable of establishing sufficient interactions to be able to carry out, and those of the binding to its ligand or other things such as catalysis. Another aspect is also recognizing the functional promiscuity of the proteins in such a way that variations in terms of function not only derive from the structure of the protein, but we also have to recognize that these variations are due to other factors such as gene expression, changes in the reading frame, post-translational modifications, interactions established between proteins, as well as the cellular context in which said protein is found. And finally now, we can precisely evaluate what the evolvability of proteins would be, in such a way that we must then recognize the contribution of this functional promiscuity, as well as the diversity of the conformations of these assemblies, where each of these could precisely be interpreted as a form of substates that would represent the entire protein and that are also subject to its choice in that sense. Obviously, depending on the physiological conditions, that could be regulated either through these mechanisms of post-translational modifications or by the concept that we previously reviewed of the polarity associated with the interactions that are established of what would be the binding site with respect to its specific ligand, which modulates more or less the flexibility that a protein has to be able to explore different evolutionary trajectories. Again, the idea here is that we can explain any biological object, in this case a cell, as multifaceted. For example, when explaining a specific cell type, we can pay attention to the way in which they were discovered throughout our history. We can also explain it in morphological terms and obviously identify if there are certain relationships, both in terms of its structure and now also in terms of its function, right? Explore what aspect precisely may get associated with these functions. In evolutionary terms, we can also evaluate what would be a set of specific genes that, being phylogenetic, can help us create some hypothesis as to how this process of cellular diversification took place, which is also extendable to what would be the world of proteins. At the molecular level, for example, when we analyze what its landscape would be from a closer neighborhood, in terms of we can now express some type of cell based on the genes it expresses, and then see why those genes are being expressed in that cell under that specific context. Or we can even cover it from what would be developmental biology and see precisely at what stages of time do certain cells present greater or lesser regulation, and whether there would be a new cell type that we may not be detecting for some reason or reason. The idea is that in order to really explain the white elephant in the room, it is necessary to integrate multiple points of view to really obtain a better image of what we intend to explain. At least in this course, it is quite clear that we are focusing a lot on what the information derived from molecular biology would be in terms of biochemistry and molecular processes. But really, if we intend to explain what life would be, some species, its behavior, among other aspects, it would be necessary to understand this biological object in terms of its genome, its physiology, its ecological context in evolutionary terms, as well as the molecular biology associated with all of this. Then really give explanations to any biological object, because it would be necessary to have this type of perspectives that are more comprehensive in this aspect. And to finish, we must recognize that we biologists also need to recognize precisely the contingent role that history plays on the entire matter of life, as well as the idea that the concepts that we use really. It depends on the empirical context in which we find ourselves, which are always changing. For example, when we were just working with what would be blue-green algae, we properly referred to them as blue-green algae, in the sense that we thought they were protists, And later we found that not all blue-green algae were actually blue-green algae, but that some of them were xenobacteria. That is, they were bacteria with the capacity to carry out photosynthesis. And so we have been modifying our concepts associated with, for example, what a species is if, for example, we have to adopt it as a more discrete perspective or a more continuous one, to be able to operate in terms of the information we have, and to be able to make more reflections on this matter. In particular, I highly recommend that you watch the series of 3 seminars that Antonio Lascano gave at the National College about what Schrödinger's contribution to molecular biology was, and whether this character can precisely be considered one of the fathers within this area. Which, as a spoiler, is a clear no. But his contribution was obviously tempting in terms of molecular biology from the perspective of a physicist. And within this series of seminars, precisely what this Antonio suggests to us is that we must understand precisely that. In order to explain life, we need to consider both physics, chemistry, and their history. Because precisely we are made of atoms, we cannot violate the physical laws that are imposed on this type of matter. But it is not enough for us to be able to explain ourselves with physics and chemistry alone. We must recognize our history in such a way that it is not so easy for us to predict history, since this is really a contingent event. For example, no one could have predicted that a meteorite would fall, and that it turns out that mammals had some success because of what would be the failure of the dinosaurs, and that now we are talking here about protein evolution. There are even some who say that we can possibly predict evolution, but only in small time ranges. But in large time ranges, this is obviously made more difficult by the end of an impossibility. Here again what Lascano precisely suggests is that life is really empirical concepts that depend precisely on the historical context in which it takes place, which we can now interpret in a more summarized way as the fact that life really is matter and with history. So, in order to understand life and be able to precisely adapt biology to our algorithms, it is necessary to look at evolution. Because we cannot understand life if we do not understand how it evolved. And from this understanding, we can derive some premises, some ideas that help us encode better algorithms to better analyze our data and interpret it in a consistent way. And as another perspective that was also quite interesting and quite recent. This is another one by another colleague called Victor de Lorenz, who presents us with this quite provocative title of the uncertainty principle applied in biology, because again molecular biology, at least in its first steps, had a great mechanistic heart, not the idea of being able to describe life only in terms of its parts, and obviously having to isolate them in order to do an experiment and characterize said part. And then to say that it fulfills such a function given that it is impossible for us to really isolate any component within what would be the cell. Then in this case, it is clear that molecular biology, at least in its beginnings, had a more mechanistic perspective on which it was built. And then the idea here that at least this Victor suggests to us is that now with what would be the applications that are had with artificial intelligence algorithms, we can now change from this mechanistic perspective in such a way that now we no longer even fully understand what is happening, but we simply put everything in an algorithm, and we teach it what precisely the answers are so that the algorithm finds the solutions, and now when asking it about some data that we do not know, now this algorithm has to give it some suggestion about a specific solution. And then this raises the idea that then this will be the end of this mechanistic perspective that was pondered at least during the first steps of molecular biology. And then again, Victor suggests to us that really, since this mechanistic perspective so far, for example, has not helped us to be able to cover all the problems, such as protein folding, an aspect that is considered resolved in terms of the contributions made by the AlphaFold algorithm. And in this case, Victor tells us that this is precisely where this type of algorithms would fall in order to be able to address other problems that perhaps we cannot solve through these mechanistic perspectives. And this is like a start for what would obviously be the next class, where we are going to address in more detail how the AlphaFold algorithm works. So we come to the conclusions, and to do so we move on to the first one. The first, evidently, is that what evolution really is operates at multiple scales, both spatial and temporal, and in each of them there are associated mechanisms that are characteristic of said ranges, which in all cases include natural selection, but are not limited only to that, in such a way that together what they do is reconfigure the diversity at each level. As a same conclusion, we have to proteinogenic amino acids, that is, those that are most commonly associated with their ability to be found in proteins, could possibly have been favored on this primitive Earth due to a combination of their bioavailability, their ability to fold into three-dimensional structures, as well as their compatibility with their other molecular neighbors at that time, such as these early genetic codes. The third conclusion is that we can see proteins like this as a kind of coevolution with the ribosome, as well as with other molecular entities, such as enzymes, metals, such that together they diversified through multiple genetic mechanisms. As a fourth conclusion, we have that the fact that a protein can have a specific function represents a genetic balance. In terms of robustness against deleterious mutations, as well as its ability to evolve, this concept is known as evolvability. And obviously both concepts, both robustness and evolvability, depend both on what the structure of the protein is, as well as the cellular context where it is found, and also obviously on its evolutionary context where it is being evaluated. As a fifth conclusion, we have that the diversification of protein functions could arise through these networks of promiscuity of their functions, which is also seen very hand in hand with this idea of polarity, where precisely there are residues that what they do is give a greater or lesser degree of freedom due to the interactions that are established with what would be the main chain of the protein together with the residues of the active site. And as a final conclusion, we have that epistasis could really be seen as a kind of limiting factor of the evolutionary pathways that can be accessed throughout the evolution of proteins, as we reviewed previously. So this is what we cover in this class, and the next one will now be to discuss how the AlphaFold algorithm works. So that's all for now. Thank you very much and see you in the next class.